Okay, so here we go back with where we left off. So the rest of the packet, which is like 13 through 18, so like 5 or 6 pages, is all about DNA replication. And so this is going to be the big thing that if you knew one thing for this quiz, it's going to be what we cover from here on out. And this is going to be a big deal for your learning of subunit 2 also. So take that for what it's worth. Down here, I didn't even circle each individual vocab word just because we have so many of them. But the vocab is going to be big. If you guys don't know what these words mean, this isn't going to make sense to you. And so this is why the homework for this section is what it is. The homework for you is going to be for you guys to do a vocab assignment. So anyway, so here we go. I'm going to, for the most part, stop following along in the notes, and I'm just going to draw for you guys. But I wrote kind of as a note to myself, we'll start here in class on, depending on which day you guys are, Monday or Tuesday, because this is where I'll start explaining. But for my online only class, I wanted to do a decent job of covering this today, and then this will give you guys a little bit of a start for our first day back. So anyways, so DNA, and this is a issue that I have a lot of people have problems with over the years. So DNA exists in what we call a 5 prime, the little apostrophe or whatever you want to call it. It's called a prime, but it looks like an apostrophe. DNA exists in a 5 prime to 3 prime strand, and then the other strand, because a double helix is made of 2 parts, it's double. The other side is 3 prime to 5 prime. So that for this reason, it's called anti-parallel. And so to come over here, so here's my double helix, and we're going to kind of get rid of the notes now. So I took this double helix and I redrew it so that it was like untwisted. Now that doesn't happen in that doesn't happen in your cells. This is just so that you can understand what I'm doing, and it's going to be a little bit easier for me to draw. So if the one side of the double helix is 3 prime to 5 prime, the other side is 5 prime to 3 prime. And if you say, well, what's the difference? It has to do with the ends of the double helix. So this end, this 3 prime end, and this 3 prime end are the same. And the same with these two 5s. And again, if you're like, well, why is it like that? We didn't make this. This is just the way that it is. So that's just something to kind of know, because we're going to talk about that later. So again, with the classic zoom in box, I zoomed in so that you can see with a little more detail. And maybe now you can see, like, so there's a nucleotide, and then there's a nucleotide, and then there's a nucleotide. And so I have this side that's my 3 prime to 5 prime chunk, and this side that's my 5 prime to 3 prime chunk. I wrote in my nitrogen base letters, or at least the first letter, so that's an adenine, that's a cytosine, that's a guanine, that's a thymine. And then I didn't write this side in. So as I do this, I'm going to have you guys tell me, which one to write here. And if you don't know, then you need to take a step back and try to figure out why. And go back in the notes and look at which ones are complementary. So if this one is an adenine and it's bonded to this one, what's it going to be? So the answer would be a thymine, because adenine and thymine are complementary. So if this one is a cytosine, the complementary base would be guanine. If that one's a guanine, then its complementary base would be a cytosine. So again, these two are the same. It's just like a flip-flop. If this one is a cytosine, that one is, we just did this one, a guanine. If this one's a thymine, its complementary base pair would be adenine. If this one's guanine, its complementary base pair would be cytosine. And if this last one is an adenine, then its complementary base pair would be thymine. Again, I'm guessing that the most of you will think that's pretty basic. If any of you are lost on that, go back in the notes, read about complementary base pairs, send me an email, get it figured out, because if you're lost there, we're going to have some issues, moving forward. So I'm going to peek at the notes. You guys don't have to look at it. I just want to make sure that I'm getting the right stuff here. So I'm going to just do some drawing, some doodling over here, so you guys can kind of see where I'm going with the notes. I'm used to doing this on a chalkboard, which may not seem like that big of a difference to you guys, but I can erase really easy on a chalkboard. I can use my hand or the eraser, and so this is going to be different. So if this is a little bit choppy, this is why I'm starting here with my in-class stuff when we get back to school. So the first thing that happens, so I'm going to redraw my double helix, my straightened out double helix here. So the first thing that happens in DNA replication is that we have a special enzyme, which you guys can remember from early in the year that an enzyme is just a protein. We have a special enzyme called a helicase, which I'm just going to draw as a little circle. And that helicase is going to come and sit on each one of those hydrogen bonds, which I'm going to pull this one back over. Remember the hydrogen bonds between the complementary bases. So it's going to come and sit on each of those hydrogen bonds and break them. So that's the role of the helicase. It breaks those hydrogen bonds between the complementary nitrogen bases. So eventually, you're going to be left with a thing that's going to look kind of like this. And I hope that you guys are following along with what I'm doing here. So that helicase is moving down that double helix, breaking those hydrogen bonds between those complementary bases. This division, it looks like a little fork in a road, like the road's going and then it splits, is creatively called the replication fork. Like I said, very creative. And that helicase will do that the entire length of the DNA molecule, which is going to be tens of thousands of complementary base pairs, all the way from the top to the bottom. So that's the role of the helicase, to break those complementary bases apart by breaking those hydrogen bonds. The next thing that's going to happen, so really while that's happening, I'm going to redraw just for the sake of my ease, as that helicase is going. So if this is the 3' to 5' end, you are going to have an enzyme called primase putting a little segment of RNA up here, and that piece of RNA is called a primer. So primase is another enzyme, and what that primer does is it's the little signal to say, hey, start making the DNA here. And so then your DNA is able to say, okay, well, if this is, let's say that was an adenine. If that was an adenine, that means that we need to put a thymine there. So it's going to take a nucleotide that's thymine and attach it, and because of that primer, it knew where to start. Again, if this is, I'd like you guys to watch the video, read the notes, maybe go back and watch the video again, and hopefully that makes sense. So that would be the very first step. And again, it's just going to depend on which bases are complementary. So DNA is being made in a 5', what's going to be to 3' direction, which is the only way that DNA can be made, because that's going to cause us a problem on this side later. I'll get back to that in a second. So I'm going to redraw over here just so there's less of a mess. So again, so this is our 3' to 5' side. This is our 5' to 3' side, but we're not worried about that side just yet. So starting right here, we had our primer. It's just a little piece of RNA. We haven't even talked about RNA yet, so just, if there's a question in your quiz, a primer is a piece of RNA. It was put there by primase, another enzyme. Let's write in some nitrogen base letters here. So that first one, I'll do it in a different color so you guys can maybe kind of see how it's being put together. So we're going to get a nucleotide that's thymine, and it's going to be attached. And then it's a new enzyme. I'm kind of, again, still following along with the notes. So this enzyme that's attaching these complementary nucleotides is called DNA polymerase. And what's an enzyme? It's a protein. So it's a specialized protein that does only this. So it attaches that one there, again, it makes that hydrogen bond. So then the next one, if this is a thymine, it's going to take a nucleotide that is adenine and attach it. It's going to move down the line. If that's a cytosine, it's going to attach a nucleotide that's guanine. And if you're like, where are these nucleotides coming from? There are billions of free nucleotides within the nucleus of your cells. So that's where these are coming from. If that one was a guanine, it's going to attach a cytosine. And notice there are some gaps here. I'm going to get to that in a second. So DNA polymerase is going to make in one continuous piece the 5' to 3' piece of DNA. And that's going to be called the leading strand. And it's called that because it gets done first. And so that'll be done in one piece, all the way from the top, thousands of base pairs all the way down until you get to the end, and then it's going to stop. Remember, because DNA can only be made in a 5' to 3' direction. So this side, what exists in our like parent strand or template strand here is the 5' to 3' side. So we would be making DNA in a 3' to 5' direction, which is a no-go. So we can't do that. But I'm going to get to that in a second. So to wrap this up, once this is done, you have another enzyme, I'll go back to a different color, called RNase, that removes the primer. It just gets, you don't really want RNA in your DNA long term, so it's going to get rid of that. And then you have another enzyme called DNA ligase that's going to attach these nucleotides. So then this side's going to be pretty much done. This side isn't done, so we have to talk about this. I need more space, so I'm going to redraw again. This is what I'm used to doing on a chalkboard. This is why. Maybe if it's not making sense to you guys, that might be why. So this is my 3 to 5. This is my 5 to 3. And like we said, this side's already done. I'm not going to redraw the letters for the bases. I'm just going to do it like that. So this side, the piece that we're going to be making would be 3' to 5', and we cannot make DNA that way. DNA can only be made this way. So you're like, okay, well, let's wait till we get all the way to the bottom, and then we'll start at the bottom and go all the way to the top. And I suppose that would make sense, but that's not what happens, so that's not what we're going to teach you. Instead, what'll happen, it'll wait till there's a long enough chunk, and it's going to be a couple hundred nucleotides long, which it's going to be like 6 here because I made you a drawing with pens on paper. But after it's long enough, a couple hundred nucleotides long, the same thing that happened up here is going to happen over here, except it's going to start in the middle and go up. So primase is going to take that and make this little piece of RNA, and it's going to attach that first complementary nucleotide, which, okay, so if that was an adenine, then it's going to be a thymine. If that one's an adenine, the next one's going to be a thymine. If that one was a cytosine, the next one that attaches is going to be a guanine. So the same thing you saw over here. So that's going to happen all the way down. That primer is going to be removed. And again, I can't erase because it's not a chalkboard, so I'm just going to scribble. And then DNA ligase is going to attach these pieces. So DNA was made in a 5' to 3' direction, just in this little chunk. And those are called Okazaki fragments. And they're called that because a Japanese guy named Reiji Okazaki figured this out, so they named it after themselves, which is kind of the thing that usually happens. So once this piece is made, and that helicase comes through and breaks more of these bonds, once it's, again, a couple hundred pieces long, you're going to have another Okazaki fragment made. And so this side is called the lagging strand, because it takes a little bit longer to do this. The leading strand just boop, 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 all the way till it's done. One piece. The lagging strand, it's got to wait and wait and wait and wait. Okay, we're going to make a couple hundred. And then it waits some more, and then, okay, we're going to make a couple hundred more. So it just takes a little bit longer for that to happen, which is why we call that the lagging strand. This video is getting a little bit long, so I'm going to actually make another video. It's going to be shorter, but I'm just not wanting to upload like a 20-minute video. So hang on, and I'll wrap this up in the next one.